Today we will learn about SAM file format. SAM means Sequence Alignment Map. Let's take an example where reads are mapped to a reference sequence. This example was taken from SAM tools documentation. In this example, there is a reference DNA sequence and coordinates. In SAM file format, one based coordinate method is used. That means the first base of the reference has coordinate position number 1, second base has 2, third has 3, and so on and so forth. In this example, 6 reads are mapped to the reference sequence. If we look into the read IDs, there are positive signs and negative signs. Plus signs mean those reads mapped to the positive strand, and negative sign means those are mapped to the negative strand of the reference DNA sequence. For the first entry, it has /1 and the last entry has /2. That means these two reads are properly paired, and this one is the first of the pair and this is the second read of the pair. Now if we look into the alignment, we can match base by base. For example, here AG has been inserted in this read 1. This AG does not exist in the reference genome. Also, G is deleted in this read. Now if we look into the 6th read, that shows this read has exact match to the reference DNA. And for other reads such as read 3 and 4, read 3 is an example of a chimeric read and read 4 is a split read. Split read or chimeric reads mean that different subsections of the read mapped to the different locations of the genome. To summarize, what we can see, the read alignment is not always perfect. A read not necessarily always mapped exactly with the reference DNA. So each read could have different alignment features, or there could have different alignment information related to each reads. So the question is how we can store those alignment information for each of the reads in a file. This is why SAM tool standardized the definition how we can store the read alignment information in a text file. Let's take a look into a SAM file. SAM file is a text file that starts with header lines. Header lines has @ sign. HD, this line contains the file label metadata, for example the version of the file format and sorting order. Here in this example these are coordinate sorted, and the second header is reference sequence directory, and the name of this sequence is ref. In this example this is ref, and the length of the sequence is 45 base pair. And all the reads that are aligned or mapped to the reference, these are stored in each of the lines in the SAM file. So SAM file has mandatory 11 columns. Each column stores specific information about the alignment of that particular read. There could have additional information in additional columns. So what those columns mean? In this table I am showing the meaning of each of the column or what type of data they store. For example, first is the queue name or the query name. In our case this is R001, R002, R003, etc. Second column is the bitwise flag information. Third is the name of the reference. In our case it is ref. Fourth is the position of the alignment. For the first read, it aligns at the position 7 of the reference sequence. So the value is 7. Then it is the mapping quality, which is followed by the Seeger string, and column 7 is the rnext, the reference name of the next mate, and the next column is the pnext, that the position of the next mate. For this read, this is a paired end read, the position of the next mate is here. If we compare with the reference, the position is at 37. So this 37 value is the pnext value, and this is the template length. That means the entire length of this pair. For example, the length from here to this second pair is 39, that is the template length. And after that, the 10th column is the sequence and 11th column is the freight quality score. If it does not exist, then star signs are used. To summarize, SAM file format has header lines and has 11 mandatory columns. These are all tab separated. Most of the columns are self-explanatory. What do they mean? What kind of data they do store? So if we look into this naming convention, we can easily interpret these columns. Also, we have shown that the examples here matches with the data in the SAM file. Now I will elaborate on the interpretation of some of the columns. When we map a read to the reference sequence, this read could map to the multiple locations of the genome. This frequently happens at the repeat regions of the genome. So we want to know the probability that the read mapping was accurate. This is the formula how mapping quality is estimated. In this example, map Q value 30 means the probability of incorrect mapping is 1 out of 1000. So this scaling is similar to the freight quality score. I have made a separate video on freight quality score. Please take a look into this video. Cigar string is the 6th column. In this column, we can see some numbers and some letters. So those actually explains the entire alignment of this read to the reference. To elaborate, these letters means, for example, M is the alignment match, I means insertion, D means deletion, N is a skipped region, and also have other information. Let's compare this cigar string with our alignment example. For the first read, there are 8 matches, 8 bases here that matched, and then 2 bases are inserted. So in the cigar string, it says 8 matches, 2 insertion, then 4 match. Let's see, then GATA, 4 match, and then 1 deletion, and then again 3 match. So this cigar string is explaining all the details of this alignment. For rest of the cigar strings, please try yourself to match the cigar string with the alignment example here. SAM file is a text file. With the alignment information of millions of reads, the SAM file size could get much bigger. Therefore, SAM file is compressed into BAM file or CRAM file. Both BAM and CRAM files are binary files that are efficient in saving storage space. There are several tools available to work with those file types. For example, if we want to convert from one file type to another, or if we want to merge multiple files into one larger file, or if we want to access the information of those SAM file or the binary versions of the SAM file, we can use those tools. So SAM tools, SAMBamba, BAT tools, these are the most popular tools used to work with SAM file. We can perform numerous operations on those files using these tools. I am making other videos on those tools. Please check out those videos as well. Thank you for watching this video. Please find out my courses on my website. See you in the next one.